Hello learners, today we are going to learn about the importance of cell culture medium, the selection criteria, and the basic components of culture medium. A very specific cell culture medium is needed to propagate different types of cells. Cell culture medium is important to maintain healthy, proliferating cells in culture. A highly pure and defined cell culture medium is a key step for biomedical application, especially for cell-based therapy. So which culture medium to use? This is a very common question among new researchers and cell culture operators. The selection of media is highly dependent on cell types and the functional characteristics of the cells. There are three ways you can actually use to get information on suitable culture medium for the cells used in your study. First, you can get it from the established protocol in-house manual used by independent research group. Second, you can get it from literature search and review from the general published protocols in journals or books. And the third one, you can get it from the company or suppliers that provide the cell lines for your research. The selection of culture medium is also depending on the stage of the cell growth, whether you are culturing the cells for expansion, making sure that the cells are doubling in number, or if you are culturing the cells to differentiate them to become committed cells, which is more applicable for stem cell population. Next is the main components of culture medium. Culture medium usually consists of basal media, which has buffer, phenol red as a pH indicator, glutamine, and nucleosides. It should also have growth factors or supplementation, such as the non-essential amino acids, antibiotics, and last but not least, serum or serum alternatives. This basal medium can either be the classical or modified type. The classical one are the chemically defined medium and named after researcher or research institution. For example, the famous one, RPMI medium, which is named after Roswell Park Memorial Institute. Each of the basal medium differ in identity and quantity of buffer, salts, and also growth supplementation. For instance, DMEM and MEM is also another type of classical medium. Meanwhile, the modified type is a culture medium that is developed over time by mixing the basal media in certain ratio. For example, 1:1 mixture of HEMS F12 and DMEM. Buffer is another important component in culture medium to maintain the pH level and provide a buffering system for the cells in culture. A carbon dioxide incubator is used to culture cells to provide it with the optimum temperature, moisture, and to maintain an optimum pH. In order to culture cells under optimum conditions, the culture media should remain at neutral pH around 7. When the media contains carbonate buffer, the carbon dioxide gas from the cylinder is let into the incubator in such a way that the pH will remain constant. Since the reaction exists in equilibrium, you can control the pH accordingly. This method is low cost and non-toxic for the cells. It is also possible to add a buffering compound to the media using sodium phosphate or trace or HIPIS, but often these compounds affect the cell's growth. Although this chemical adjustment is more efficient to maintain pH, it is not carbon dioxide dependent. Controlling pH this way could be toxic to the cells and also a bit expensive. Next is the culture medium supplementation. Growth factors is needed in culture medium to stimulate glucose transport and utilization, as well as induce uptake of amino acids. In most cases, growth factors are needed to induce differentiation and maintenance of cell commitment. Ideally, growth factors should be freshly added into the medium for an optimum effect. Another important component in culture medium is the non-essential amino acids, which are commonly synthesized by the cells. It is crucial for cell division and growth. However, it will still be provided in culture medium to avoid potential of nucleic acid depletion. For many years, serum has been added into the culture medium, which aids for cells attachment, binding, and neutralization of toxins. However, the use of fetal bovine serum also comes with few disadvantages, such as risk of infectious agents, variable composition between batches, high cost, undefined growth factors and hormones, as well as issues with the regulatory bodies. The serum must be properly inactivated before use at 56 degrees Celsius for 30 minutes to destruct complements and immunoglobulin, and also if there is any viruses. However, overdoing the process of inactivation may as well damage the growth factors, hormones, and vitamins in the serums. Apart from using fetal bovine serum, researchers now can opt for serum-free, xeno-free, and animal component-free medium. Each of these serum alternatives media provides chemically defined components, either synthetic or made of recombinant factors. The use of serum-free medium for cell culture work has increased on the definition and purity of the culture medium itself. It helps to produce consistent performance between batches of medium, thus producing more reliable experimental results. Other than that, it also offers more control over cell environment and practically improves the evaluation of cellular response. The medium used for cell harvesting are important, especially among the adherent type of cells. Trypsin or acutase was used to cleave protein bonding between cultured cells and the flask or dish surface. The use of this enzyme will allow for resuspension of adherent cells for cells harvesting. The optimal temperature for this solution to work is at 37 degrees Celsius. In some formulation, EDTA might as well be added as a chelation agent which binds to calcium and other ions. Sometimes, collagenase might also be used on tissues that are too fibrous during the isolation step or cells that are too sensitive to trypsin. Apart from growing the cells for expansion in the lab, sometimes the experimental work can also be performed to stimulate or test the cells within different growth conditions. For example, different conditional medium is needed to stimulate glucose transport and utilization in the study of pancreatic beta cells. Researchers can also use conditional medium to maintain the cells for a very specific differentiation capacity. Also, certain conditional medium might as well be used to direct migration of cells for in vitro disease model, cellular response, and signaling studies. That's all for now. Thank you very much for watching.